Hi, my name's Sarah Comer, and today I'm presenting some preliminary results from my part-time PhD with the University of Western Australia, studying the ecology of the feral cat on the south coast of Western Australia. This work should be submitted sometime this year, I hope. I'd like to acknowledge the traditional owners on whose country this work has been carried out, and on whose land we are gathered today, the Wadjuk people. I pay my respects to elders past, present and emerging. I'd also like to acknowledge my many DBCA colleagues who've been involved in this project, my new employer, Bush Heritage, and my supervisors, Peter Speldwin, Dale Roberts and Dave Algar. So a bit of background, this project builds on the work done through the South Coast Threatened Fauna Recovery Project, which you've heard a bit about today from Abby Thomas, who talked about Kyloring and efforts to conserve this amazing parrot. So the work that was done to conserve Kyloring was using that species as a bit of a flagship for conservation, recognising that the south coast of WA is a hotspot for vertebrate conservation, and there was, a number of significant mammals and birds and other species across the tenure that's part of of my project. So from Cape Arid and Neutzland in the east to the Fitz-Stirling River National Park, Two Peoples Bay Many Peaks, and then the Fitz-Stirling landscape, which includes areas managed by Bush Heritage, DBCA and other partners. And this project sort of builds on that idea that the management of cats in large unfenced landscapes needs good knowledge and understanding of what cats are actually doing in this part of the world. So the aims of my project were addressed across the four areas, and specifically looking at diet and increasing our understanding of the spatial and temporal pressure cats are putting on native species, and looking at the potential to use stable isotopes to support traditional dietary analysis. To look at spatial ecology of cats and how this could be used to inform baiting and control programs, and to investigate the potential of prey availability to be predicted using some remote sensing techniques, and with a view to informing the timing of baiting in the areas that are subject to aerial baiting programs. So some preliminary results from the dietary analysis. So this is output from 197 stomach content taken from cats across the four landscapes. I looked at the standard metrics, including frequency of occurrence and index of relative importance, to allow for a comparison, and some of the output from that is in the figures down the bottom of this slide. 58 native species, 17 mammals, 28 birds, 13 reptiles and 8 invertebrate orders were identified from stomachs. 10 of the species detected were conservation dependent, and that included both mammals and birds. And while native mammals dominated the diet of feral cats from the reserve areas, house mice were the most frequently consumed prey in the Fitz-Stirling. The stomach contents were then used as a baseline for stable isotope work, and that this included both looking at niche and whether these methods had some value for mixing models. So I did some exploration into seasonal niche shifts, and you can see some of one of the figures from this work in the bottom left-hand corner, which just basically looks at the niche width over seasons in Cape Arid, Fitz-Stirling River National Park, and the Fitz-Stirling in the middle, and you can see a much broader niche, isotopic niche in the Fitz-Stirling, which interestingly reflects what was being seen with the diet from stomach content analysis. I also investigated the scope for Bayesian mixing models to be looked at to determine long-term temporal changes in feral cat diet, and I only did this work in Cape Arid, where there was a relatively consistent diet, as you can see in the niche figure, which Cape Arid's actually the far left-hand corner of that plot. And the two other figures on this slide are the isoscape plot, which shows the 5 collapsed food groups and the feral cat isotopic signatures, and then some output from the mixing models in the form of posterior plots, which show general priors in the top and informed priors in the bottom. There's still a little bit of work to do with these to actually ensure that they meet convergence. But interestingly, the preliminary results are suggesting that in Cape Arid, the mixing models are supporting the stomach analysis, but also finding evidence of other species in feral cat diet that aren't being picked up in the last meal. So there's a bit more to do in that space. Moving quickly onto the behaviour and spatial ecology. So this was looking at GPS collar data from 47 cats that were trapped across the landscape, as you can see in this map. This is just the males. And I used continuous time movement models in the package CTMM to establish if feral cats were resident, and then to look at the broad trends in movement patterns and home range size and the utilisation distributions across these areas. The variograms in the bottom left-hand corner of this slide just show an example of range residency being met, but with some fluctuation in that displacement, which is likely associated with lunar periods. And the telemetry data and CTMM was also used to look at some resource selection models, and there were significant selection for cats for moisture gaining features, which included creeks and river systems, but also granite outcrops and wetlands. So there's positive selection in Cape Arid and the Fitz-Stirling and the Fitzgerald River National Park. Terrain ruggedness wasn't significant over the entire data set, but when pulled apart, there was certainly signs that cats were moving into more rugged or heterogeneous terrain during their resting periods. Time since fire was a negative selection, so younger fuel age in Cape Arid National Park, and then positive selection for productivity in Cape Arid, the Fitz and Two People's Bay Many Peaks. The data from these collars was regularly sampled, which is why I looked at resource selection models rather than step selection functions. And I think the spatial ecology, if you look at the hourly displacement for regular movement patterns, this and the strong preferences for different habitat types can be used to inform more targeted or intelligent baiting programs. Quickly, I'm just going to touch on the prey availability. So this was just looking at whether a long-term regular small mammal trapping program could be used to predict when baiting might be optimal to increase the probability of a cat encountering a bait. And this was a 3-year trapping data set, and basically I used dynamic habitat indices and other climate parameters and found that there was a strong significant relationship between biomass derived from EVI, or Enhanced Vegetation Index, and also a significant relationship between trap success and rainfall in the preceding 12 months. So potentially some simple parameters that can be extrapolated across larger landscapes to help be a little bit more targeted with when we deliver baits. And that's pretty much the summary of the work that was done in a very short, snappy talk. But yeah, hopefully, once this work's written up, we'll have an increased understanding of the species at risk and some simple tools to build a more comprehensive picture of feral cat diet over time. Also, some metrics and tools that can be used to inform both temporal and spatial delivery of baiting programs at a patch scale and increase increase the cost efficiencies, and a lot of learnings that demonstrate the need to be both adaptive and integrated across tenure boundaries, as I mentioned before. So yes, very quick, lots more to come, and I would just like to thank the amazing team of colleagues and friends at DBCA who've been involved in this work throughout, Bush Heritage and DBCA's Invasive Animals Program for funding some of the work, and everybody that's been involved. Thank you.